Hello, I'm Brian Kinghorn, and this is the first video in a series on genetic markers. So we'll talk about individual genetic markers and managing them. These may be markers for genetic defects or quantitative trait loci. We'll also talk about managing suites of genetic markers, potentially hundreds of them, and also we'll look at genomic relationship matrices and how we manage these in Matesel. So this first video is on coding genetic markers in your main data file. So this is the example Matesel classic demo, and you will see some of the columns. I've got these rather unusual headers which represent genotypic information. So we have two types. We have those starting with a little g for genotype, and that's where the genotype of the individual is known for that locus. And we've got two loci like that. And another two marker loci are each represented by three columns. That's because we need to represent them by probabilities of being of each of the three genotypes because we don't have perfect information. So let's look at coding header for a genotype. G tells us it's a genotype. 2 tells us the number of alleles at the locus, and in animal breeding that's almost invariably 2, but we can handle more than that. 3 says that we're considering 3 different genotypes at this locus, and that's the 2 different homozygotes and the heterozygotes combined to give us 3 classes, and that's the most common format for animal and plant breeding data. This is the name of the locus. Putting a dash at the end of it for marker 1 means that when we show a representation on the interface, then it's more clear what's the marker locus name and what's the name of the allele or genotype that's being considered. 1 and 2 are the names of the first and second allele in this case. Here's another example. AM is, I think, curly calf in cattle, and our allele names can be plus and D for defective potentially. But in this case, rather than 3, we've got 4 genotypes represented, so that we could have the 2 homozygotes, but also the heterozygotes separately represented. So if you have that information separately represented in your data, you can present it in a Matesel data set. So you'll notice that we can have more than just 3 or 4 columns. We can actually have 5 for the allele frequency in the top row, and in lower rows where we're looking at genotypes, we can have 4 different columns. So that is because we have 3 genotypes in this case. When we mate a 1-2 with a 1-1, then the progeny in that family can either be 1-2 or 1-1, and the average frequency in that family is therefore 75% 1. And when we look at this, we can see we have potentially 5 different levels of allele frequency and 4 different levels of genotypic frequency. We can't have 0.75, and I've tried to get it here, but it doesn't exist. And you can consider this table and you'll see that a 0.75 cannot be had here. So that's why we get a number of columns greater than 3 in our output. So now let's look at the genotype columns and see what are the entries that we put in there to represent the different genotypes we've got. This is how it works, and you can see this in our manual on page 19. So if we have the alleles plus and D, then for a 2 allele 3 genotype example, then the 3 genotypes are the 2 homozygotes and the heterozygotes collectively, and these are represented in the data file by 1, 2 and 3. On the other hand, if we were to have 4 different genotypes, because you have data separating the 2 heterozygotes, then 1, 2, 3 and 4 should be used according to this. So that's plus plus and DD, but you may be using 1, 1 and 2, 2 if your allele names are 1 and 2. So even if you're just using 3 genotypes represented in the data file, Matesel will separate the heterozygotes automatically under all circumstances and give you separate output for the 2 directions of the different heterozygotes. And that could be useful to you because you might have imprinting, for example, and there's some desire to have a certain direction in output in your heterozygotes, and Matesel will handle that for you. That's us finished with the genotypes. Let's move on to genotype probabilities. So in some cases, we do not have knowledge of the genotype of an individual, and we can infer it from the genotypes of other individuals or other information such as phenotypes. So in this case, we know that this individual here in the second row must be big A, little a, because it must have received a big A from its mother, and it has given a little a to one of its offspring. And so here it is, the male in row 2 is 100% chance of being a heterozygote. For the other 2 question marks, it's not so straightforward, and we use segregation analysis to calculate probabilities for these individuals. And here's an example of using the program GeneProb, spider syndrome in sheep, with in this case 6 affected individuals. And in the data file, spiders have got a 100% chance of being the affected genotype. And the result from the segregation analysis tells us the probability of being a carrier and the probability of being homozygous normal. So it's quite simple to do this. We have a penetrance matrix, which in this case is showing a direct relationship between what we observe and the underlying phenotype. So this is probably DNA information. And all we need is a pedigree and a note of what the genotype is. And we use a code of 9 for unknown. The result from that is to give us the genotype probabilities for the 4 genotypes. It's 4 genotypes because we've asked for it here in the header. And you'll see that the probabilities can differ for the 2 directions of heterozygosity, and that Hein here has got to be a 2-1. And if you look at this pedigree and think about it, you'll see that that is inevitable. And given that these probabilities are essentially continuous numbers, you will see that for the 3rd and 4th marker, we're not restricted to 5 or 4 different column levels. We have got more or less continuous values for the genotype probabilities in progeny, which are the average of the probabilities in the parents, in fact. So now, how do we code? Well, let's just again look at the genotype one we've already done. So that's very similar. I'll move now to the genotype probability, and you see the differences. We've got a GP, but there's also now an extra component here. This 2 tells us the genotype number. We are going to have a column for each of the genotypes for a certain marker, and this tells us which of those columns it is. So with 4 genotypes in our example, and here they are, we'll have 4 fields in the dataset containing genotype probabilities, as we have here. So let's look at those 4 headers. They are 1, 2, 3, and 4, representing the 4 genotypes. And the probabilities for any one individual will sum to 1 across those probabilities. And that means there's redundant information, and what we can do is we can omit one of those from the data set, and it has to be the first one. And what Matesel will do is calculate the probability for the missing genotype as what's left to go up to 1. And we can see that here in this data set, we only have genotypes 2, 3, and 4, and the first genotype is inferred from what is left to go. And this can save quite a bit of space where you've got rather many markers. Well, that's essentially the end of this video, but I'll just conclude by saying that if you're making simulated data sets to test many of these things, then not only can you simulate your data, but you can generate the headers in a format something like this, so you can generate your data set to put into Matesel automatically without having to do these things manually. And so here's the headers being generated, and here's the information going into the columns. Well, that's the end of this video. Thank you for listening.